afternoon. All right. What I'm going to do today, just short, make it about maybe 20-minute video, 25 tops. Go over the first of your content, the material that will be on the first quiz, first exam. Where we'll start with the lecture. Also goes along with the lab, at least initially. A lot of times our lecture and lab go completely different directions, but right now we're doing both topics in lecture and lab. We're going to do a couple of short lectures on embryology. Normally in this semester when we're in person, I go for about 2.5 hours on embryology, so it might be a little shorter. Anything I need to wrap up, I'll wrap up in person next week. But embryology, also called animal development, it basically, we're specifically looking at sexually reproducing species. Admittedly, all animals sexually reproduce, but a lot of them asexually reproduce. We're not looking at asexual reproduction or development, although it has a lot of similarities. So we're just looking at development in sexually reproducing species. And development is basically, you start at that point where the sperm and the egg fuse. That first cell of a new individual is genetically distinct. It's usually called a zygote. And you basically watch that develop from the zygote until you have an adult form. Now, we're not going to go that far. We're basically doing fairly early development. We start, actually, we'll go a little bit before development. We'll start with a little bit about fertilization and the gametes, the sperm and the eggs. And we'll go through just basically until some of the organ systems start to form. Nervous system, heart, a couple internal structures. We stop there. I don't do later stages of development. We don't have the time, and all of our our specimens tend to diverge at that point. And as we're going over development, we're going to kind of do a little bit of a comparative approach. We're going to be using 3 different species as models. We will use Echinodermata. Echino is a phylum, Echinodermata. This is an animal phylum that includes sea urchins, starfish, sea cucumbers, brittle stars. We'll use those in lecture, and this is the one that's important because we'll also be looking at this one a lot in lab. So we'll be looking at in vitro fertilization with echinoderms. So that's one group. We'll look for fertilization through early development. The second group we'll look at, I'm just going to call them that's not really a tax, it's amphibians. It's a class amphibia. This includes your frogs, your toads, your salamanders, caecilians, which aren't common around here. Those are the amphibians. Those critters, they can live on land. They're typically tetrapods, except for the caecilians. And they breed, but they breed in water, or at least in a non-desiccating environment. And the third group we'll look at, eggs, which are birds. Now, these are kind of classic examples for development, in part because all 3 of these, it's really easy to get the eggs and study them in the lab. As we'll see with the sea urchins, the echinoderms, you can grab those out of the ocean, inject them with potassium chloride, eggs and sperm come out. It's all external fertilization. So you can put the sperm and the eggs on a slide or a petri dish. You can watch the fertilization. The embryos develop just floating around in the seawater as part of the plankton. So you can watch the development. Easy to get the eggs, easy to observe them. Amphibians, same thing, external fertilization. You're familiar with frog eggs. They lay little eggs in a puddle or a creek or a pond. Males fertilize them as they're released. And again, you can get them at fertilization, watch them develop, externally in a petri dish or something. It's easy. Chicken eggs, we'll be looking at chicken eggs. They're easy to acquire, not quite as simple because, of course, they develop inside the egg. You got to kind of crack the shell off and put a window on it to watch it a little bit. It's a little trickier. What you'll notice is we're not looking at any placental mammals, which would include humans or placental mammals, where the embryo develops in the uterus. They're technically way too difficult for us to mess with. A lot of the early work in embryology was not done with placental males. It's technically too difficult. Now, what we'll notice as we go through, echinoderms and amphibians, while there are some differences, both external fertilization. The amphibians are vertebrate, but they lay their eggs in the water, so they're in an aqueous medium. Aves are going to be important because they're in a different lineage. It's called the amniote lineage, which is the same lineage as a placental mammal. So these are a model, at least partly, for placental mammal development, human development. They follow almost all the same developmental patterns. What we'll see later, I will mention placental mammal, some aspects of placental mammal development. but only briefly. There's some differences when you stop laying eggs and you let your embryo develop in the middle. Some structures are modified for slightly different purpose. But these are the three groups we'll look at. And like I said, all fairly early development. Now, in the chicken embryos, we will look at some of the structures that develop later in the lab, eyes, ears, brains. But for the echinoderms and amphibians, pretty much early development, and we stop once the nervous system forms. So these are the three groups I'll cover in lecture, and are also the three groups we'll cover in lab. You look at some prepared slides for these and these. For the echinoderms, we'll watch them actually develop under a microscope slide. Now, what we're going to do actually is I step back a little bit from development, because development starts at conception. You form that zygote, that first cell. We're going to look at the gametes a little bit first. Those are the reproductive cells. In animals, that would be sperm and the eggs. Just mention a couple things about them and look at the process of fertilization. So when I'm done on this video, I'll actually attach at the end a narrated PowerPoint and go through some of these steps with better diagrams than the nasty stick pictures I can draw on the board. But let me just mention a couple things about sperm. There's my picture of the sperm. Sperm and eggs are genetically equivalent. And what's interesting about both of them, in mammals, in humans, most of your cells are produced by mitosis. I know last semester you looked at mitosis and meiosis, you looked at G1 and ES and G2, and then the different stages, prophase, metaphase, anaphase, telophase, and what was going on. But the big difference between mitosis and meiosis is what happens into the chromosomes. Remember, most cells produced by mitosis, which are all your somatic cells, every cell in your body reproduces by mitosis except for your reproductive cells. And in mitosis, it's just cloning. Everything's identical. The original cells, I gotta check, make sure how much of my board's on the screen. Yeah, just barely made it. Most somatic cells, think of these as regular body cells, everything except sperm or eggs. Somatic cells in humans have 46 chromosomes. You remember they're arranged as 23 pairs, so you got 23 homologous pairs. When mitosis occurs, you go from a cell that they refer to as being diploid, you have the homologous pairs of chromosomes, 23 pairs, total 46. After mitosis, the resulting daughter cells also have 46 chromosomes, and the next year have 46, 40. So they're identical. Genetically, there's no changes. We use mitotic divisions to produce every cell in the body except the sperm and eggs. We do not produce sperm and eggs by mitosis. That's meiosis. So meiotic divisions in a human, in placental mammals in general, meiosis only occurs in the cells in the gonads, the sperm, to produce the sperm or the eggs. And if you recall, and I know you've gone over it, but meiosis is a reduction division. When cells are produced by meiosis, a mature sperm only has 23 chromosomes. They lose half of them. You remember the 23 homologous pairs, you separate the homologous pairs, so each sperm only gets 23. And of course, you may recall that all the sperm are genetically a little bit distinct because of the random independent assortment of those homologous chromosomes and also crossing over curves and all of that. So you have millions and millions of different sperm genetically. But they're haploid, so they're a little bit different. Now, why are they haploid? Well, let's say they weren't. Let's say you decide to go ahead and make sperm using mitosis. Could you make sperm using mitosis? Well, yeah, you'd end up with this sperm, it's got its head, its midpiece, its tail, but it has 46 chromosomes. If you made the eggs and the sperm, here's an egg. I'm using human chromosome numbers. If you produce the sperm and the eggs by mitosis, again, mitosis, everything stays the same genetically, they both have 46 chromosomes. When those things fuse, when fertilization occurred, Junior here, J. Harper Jr. would have 80, 92 chromosomes. That will not work in a male. You can't have polyploid individuals. You got to have 46 chromosomes. You have extra chromosome 21, causes Down syndrome, a lot of developmental problems. If you have a whole extra 46 chromosomes in mammals, most species, it won't develop. The embryo will just die. Now, there are a few species, a lot of plants, and a few amphibians that you'll see polyploid individuals. Sometimes they can get by, because plants in particular can self-fertilize. But the problem with that is, if you had two individuals, let's say Junior here, oops, I left the egg. Let's say Junior here with 46 chromosomes grows up, wants to have kids, he'd have to find someone from his generation but also had 46 chromosomes, and when those sperm and eggs fertilize, again, if they're produced by mitosis, or you've got 92 chromosomes in their offspring, it won't work. Pretty soon you need a nucleus the size of a basketball because you're doubling the genetic material every generation. It will not work. So we do not produce sperm or eggs by mitosis. They are produced by meiosis. So they are referred to as being haploid. Again, haploid is half the normal chromosome number. Your somatic or body cells are all diploid. They have 46 chromosomes. That is, chromosomes arranged as 23 pairs. Sperm, no pairs, just 23. So these are produced by meiosis, but sperm are also different in another way. Again, you looked at meiosis genetically, you looked at the nucleus and the changes that occur during meiosis. But during spermatogenesis, when you're actually producing the sperm are the smallest cells you'll find in a mammal. They're really tiny. Part of the reason for that is during meiosis, not only do you reduce the chromosome number in half, but sperm will shed almost all those cytoplasm. So I'll show you this on a micrograph on a PowerPoint in a bit. But basically, what they call the head of the sperm, a little bulbous bit at the end, it's basically a nucleus. It's kind of like a nucleus on a stick. There's not much to it. All you have at the head of the sperm is the nucleus containing the genetic material, and at the anterior end, there is a small structure called an acrosome. We'll refer to that a little later when we look at fertilization. The acrosome has a mixture of a lot of hydrolytic enzymes in it. These are going to be necessary to erode some of the outer layers of the egg, so the sperm cell membrane can contact the egg cell membrane. Then the only other thing you have, so you got this little nucleus on the head, a little acrosome. Again, a nucleus on a stick. There's a little mid piece or body that contains mitochondria. Of course, those are necessary to make ATP. The ATP is needed because you have this long flagellum, which using the energy produced by the mitochondria, the ATP, the flagellum can thrash back and forth. And of course, sperm are mobile. They swim. If it's external fertilization, like the sea urchins, they swim around in the ocean until they chemotactically attracted to an egg. They find the egg, they fertilize it. In a placental mammal's reproductive tract, they swim up the female reproductive tract again until they encounter egg. So they're very tiny cells. The other reproductive cells, the female gametes, are either called an oocyte or you can call it an egg. It's the same thing. Egg and oocyte can be used interchangeably. Or sound fancy, use oocyte. Egg is fine. They are, as I mentioned, genetically equivalent to the sperm in that they are also produced by meiosis. They are haploid, half normal chromosome number compared to your somatic cells. But what's also interesting to note, eggs are enormous. The yolk in a chicken egg is technically a cell. They're huge. Even human eggs, well, they're not as big as a chicken egg. Human eggs are pretty big. They're usually described as being about the size of a period on a printed page. You can pick them up with forceps and you can see them with your naked eye. It would be the same with the sea urchins. You can barely make them out with your naked eye. You can resolve them. Sperm, no way. You need pretty high power magnification just to see them. So they're big cells. They're also produced by meiosis, but meiosis as far as the nucleus is concerned, but the way oogenesis or formation of the eggs occurs is a little bit different, not genetically. So you start off with a parent cell, with its 46 chromosomes. And if you remember, meiosis goes through two divisions, right? You got meiosis 1, meiosis 2. Now, when meiosis 1 occurs, I'll just write it, meiosis 1. Of course, you reduce the number of chromosomes, you'll be at 23, I believe, at this point already. But physically what happens is, when this first division occurs, it's unequal as far as the cytokinesis is concerned, the actual division of the cytoplasm. One of the daughter cells in meiosis 1 is huge. It gets all the cytoplasm. The other one is really tiny. In fact, these are usually called polar bodies. They are genetically equivalent to the egg, but they have no cytoplasm. They won't develop. Even if they're fertilized, they're just going to degenerate and die. They have no raw materials, no energy. They're dead. Now, meiosis 2 will occur. And of course, each of the cells produced by meiosis 1 potentially will divide, including the polar body sometimes. but it'll just produce two tiny polar bodies. Over here, the big cell produces a polar body and one big cell. So again, meiosis, you produce 4 daughter cells, 1, 2, 3, 4. 3 of them are polar bodies. They aren't going to do anything. They ain't getting cytoplasm. They're just going to degenerate. That is the oocyte. Part of the reason it's so big, it gets all the cytoplasm from all the other daughter cells, so it's large. That cytoplasm is important because that contains all the raw materials, building blocks, nutrients, energy needs for the early development of the organism. So they are quite large. Now, what I'm going to do next is we'll look at some of the steps of fertilization. Mostly we'll look at fertilization in a sea urchin. That's what we're looking at in lab. We'll actually observe it. It's very similar in frog eggs, not much difference. And it's basically the same even in a human egg. There are some subtle differences. Of course, they studied placental mammals later, so some of the terminology and names for some of the structures, particularly the oocyte, are a little different. Don't worry too much about it. Mostly I'm going to go through fertilization in the sea urchins, and I'll just mention a bit about fertilization in placental mammals. There's a couple of differences, but the basic process is the same. Excuse me. I'll do that using a narrated PowerPoint because it kind of needs some pictures. I may be back to the board in a minute.
Alrighty, what I'm going to do is go over some of the PowerPoints, just kind of point out a few of the parts, go over fertilization and the gametes using slightly better pictures than the stick pictures I can put on the board. Now, what you're seeing here, this whole thing, this is a sperm, the male gamete, and just a couple parts to note. It is very tiny. This is an electron micrograph. It's pretty high resolution, high magnification, looks pretty cool. But this part here, this is the head of the sperm, labeled right here. And in the head, most of this space is taken up with a nucleus, which contains the DNA. Sorry, I'm drawing with a mouse. I can barely draw with a pencil. This looks, yeah, like a third grader did it, but no, a 3-year-old, not a third grader. Don't insult them. And then at the anterior end, that is where you'll find a little membrane-bound organelle called the acrosome. And as I mentioned in the video earlier, that contains some hydrolytic enzymes that are going to be important for fertilization. Then you got your little body or mid-piece right here. That has some mitochondria in it. It's kind of the energy-producing portion of the sperm. And then you have the big, long flagellum, which, of course, sperm are motile. They'll swim through the environment to reach an egg. Again, very small, and they are haploid. This is a diagram of an oocyte and some sperm and some other material. It's not a picture, but it works a little better than a picture, and certainly better than my diagrams on the board. Now, what I want you to note, this blue part here, the edge there, I'll sort of follow along, that is the oocyte itself. So all of this is the oocyte. Its plasma membrane is right here. And of course, it is a large cell. It's also haploid, 23 chromosomes for a human. Again, the chromosome number will vary by species. 6 in a fruit fly, 104 in a goldfish, but we'll use humans as an example. But what I want you to notice, one, it's big. They got some sperm here, kind of for scale. Here and here, and all these little tails sticking out here are actually sperm flagella. But note there's a couple of layers outside of the egg's plasma membrane. Now, this particular oocyte happens to be from a placental mammal, so the terms are a little different. But you'll notice this kind of yellow shaded bit here. This will be named the zona pellucida in the placental mammals. If this is a sea urchin or a frog egg, it's the vitelline membrane. So two different terms for more or less functionally the same thing. It's a protein containing sort of coat or layer around the egg. Then out here, you have another layer. Now, if this were a frog or a sea urchin, you've seen frog eggs in a puddle, right? They have that jelly around them. It's literally just called the jelly coat. Now, this is a placental mammal, so this isn't jelly, but functionally it's the same. They're calling it the cumulus. These are actually cells that were in the female's ovary. They adhere to the outer edge. So the cumulus cells in the placental mammal are functionally going to be equivalent, as far as fertilization is concerned, to the jelly coat of a frog egg. Now, for fertilization to occur, one of these little sperm has to get through these outer layers, because for fertilization to occur, the sperm cell membrane must contact the egg's plasma membrane. And you got all this other junk on the outside. You got the jelly coat or cumulus, you got the vital envelope or zona pellucida in placental mammals. Those must be removed before the sperm can contact the egg. Now, the way they are removed, as we'll see in a bit, it's that acrosome. So let me go to another slide. All right, now here, this is showing you an egg over here. Kind of goes from here back to here. This is the outside of the egg over here. This is the egg cytoplasm here. So this brown layer right there, that is the egg's plasma membrane. That's what the sperm must contact. Then you'll notice you have this layer here. It's kind of green and blue. That is the vital envelope. It would be the zona pellucida if it were a placental mammal. This is actually a frog egg. And then out here, there's a jelly coat here. It's a little hard to see on the slide, but that's a jelly-like layer on the outside. And here's your little sperm right here. So you release a bunch of sperm, they're attracted to the egg, chemotactic movement. They move towards sort of a chemical gradient leading them to the egg. And then they reach this outside, boink, boink, boink. They can't get in. That's going to be a bit of a problem because that sperm cell membrane has to come into contact here, and you got all this junk. So up here, kind of the anterior end of the sperm, that is the acrosome. Now, what the acrosome is going to do when this sperm contacts these outer layers of the egg, the sperm undergoes what's called an acrosomal reaction. Basically, the acrosome ruptures. Now, there's a couple weird things here, and I'll use humans as an example. Sperm that are freshly released, freshly ejaculated sperm, if you put them right in an egg, nothing will happen. The membrane is actually too rigid. If you remember from membrane structure last semester, you got the phospholipid bilayer proteins, and you have cholesterol. Sperm, when they're first released, have a lot of cholesterol, which hardens the membrane, and it will not undergo an acrosomal reaction. It can't rupture this anterior sort of tip here where the acrosome is. Sperm, actually, they call it, have to be capacitated. Sperm have to swim through the female reproductive tract in the case of placental mammals. And that process of swimming actually chemically erodes the cholesterol out of the membrane at the tip of the sperm. And so once it's made that path, swam through the female reproductive tract, it's then considered capacitated. The membrane is more fragile. When this sperm contacts the egg, the acrosome ruptures. And these acrosomal enzymes out here, they'll be released on the surface of the egg. They basically digest it. They digest the jelly layer. They'll help digest the vital envelope or zona pellucida. So they erode the outer layers of these around this egg. Now, one thing, if we took one sperm and put it in a little petri dish with one egg, plop the sperm in, assuming it's capacitated or chemically treated so it's ready to go, would it fertilize the egg? Probably not. The likelihood is fairly low, in part because these sperm are really tiny. How much acrosomal enzyme do they have? Little teeny bit, not much. And that enzyme is going to get neutralized. It'll diffuse away. So one sperm may just only erode partway through these outer layers, and that's it. It's done. That's partly why, in a human, ejaculate contains, something like 400, 500 million sperm. I read that somewhere. I didn't count them. So there's a huge number, and collectively they slowly erode these outer layers here. And over time, enough of them will erode away till eventually one sperm will contact the egg's plasma membrane. Now, here another weird thing happens. What do you think would happen if we took, say a human egg, and we took, hamster sperm, and we put them together in a little petri dish, and a hamster sperm swims towards the egg? Would it fertilize the egg? You'd have like a hamster boy hybrid or something that you see in the magazines at the front of the checkout line. Would it happen? The answer is no. But why won't it happen? Well, again, if you think of most species, now we have internal fertilization. So, you kind of figure we'd know what kind of sperm is getting in our reproductive tract, but most other species do not. Think of the sea urchins we're looking at, clams, coral, worms. They don't copulate. They don't have internal fertilization. At breeding time, they just release all their sperm, and the females release all their eggs, and they're just floating around in the ocean. If you go to some of these coral reefs at certain times of year, most of these species will synchronize their breeding to overload the predators, so all the eggs don't get eaten. But it looks like soup. You might have all the echinoderms and many of the species of coral and many of the marine worms all releasing their eggs and sperm at the same time. It's like a soup. All these different species of sperm and eggs, swirling around. And if you're a sea urchin, for example, do you want clam sperm to fertilize your eggs? The answer is no. Why not? Because If clam sperm fertilized a sea urchin egg, they're not genetically compatible. The egg is dead. It's not going to develop. So evolutionarily, you've wasted all that reproductive effort. All that energy you put into the egg is gone because some silly clam sperm fertilized your egg. Don't want that to happen. So to prevent this, most species have a species-specific recognition signal, which will only allow sperm of the right species, or at least a really closely related species, to fertilize your eggs. And on this diagram, if you look at the tip of the sperm right here, they're kind of showing me these little yellow ballies. It's labeled right here. There's a membrane protein. It's called a binding protein. Again, protein structure is genetically determined. So a particular species of sperm, let's say, frog sperm here, is going to have a particular structure of this binding protein. The egg of the same species will have, right here, will have an appropriate binding receptor. Not very ingenious names, but they work. The binding receptor is on the egg. It actually projects out through the vitelline envelope here. That's a protein. It's genetically determined. So kind of like a lock and a key, you have to have a compatible binding receptor and a binding protein on the sperm. When that protein encounters the binding receptors and they bond, that allows the sperm to contact the egg cell membrane, and they're showing you right here, release its genetic material into the egg. Yeah, that's terrible writing, but that's the idea. Into the egg. If it's a different species, it won't bind. Now, it is kind of cool, though. Cool or freaky, depending on how you look at it. They have done experiments where they'll take a human egg. If you strip off that outer layer, chemically remove the zona pellucida, and then introduce hamster sperm, it will fertilize the egg. No hamster boy, though. We're not genetically compatible with a hamster. So that sperm gets in there, fertilizes the egg, and that's it. At this point, that is fertilization. Now, do note, about the only thing the sperm contributes is a genetic material, DNA. In some species, some of the centrioles are also, paracentrioles, I think, is introduced as well. But for most species, it's just the DNA. None of the mitochondria, none of the other organelles. So the only male contribution for most species is the genetic material. None of the other stuff. If the sperm have it, it just degenerates. It doesn't end up being part of the zygote or the embryo. That is fertilization. Now, the next thing we'll look at, that sperm's fertilized the egg. This cell now is at the diploid number. If it were human, it would be 46. Keep in mind, though, you still have maybe tens of thousands of sperm out here still trying to gain entrance into the egg. I'll just put 10,000. It's a lot. If a second sperm fertilizes the egg, you have a polyploid individual. The embryo won't develop. Again, wasted your reproductive effort. This is something called polyspermy, where more than one sperm fertilizes an egg. You have to prevent that, at least in an evolutionary sense. Now, in lab, when we're doing in vitro fertilization, you will get some polyspermy because I kind of overload those sea urchin eggs with way too much sperm than you find normally. It overloads the system. So what we'll look at next is what's called slow block and fast block to polyspermy. These are methods to prevent more than one sperm from fertilizing the egg. Now, this next slide, this is not really on the PowerPoint. This is on Blackboard. There will be a little document. It says C Notes Development. All this is, I stick these on Blackboard. Use them if you want. But this is basically a list of the term and some basic concepts that you have to know. These are ones, if you just memorize these and you know these pretty well, you should get at least a C. That's why it's C Notes. You still need some understanding of processes and a little bit of a deeper understanding of what's going on. But this is kind of it. So like, for example, some of the terms I've already gone over. What does haploid mean? What is diploid? I think I mentioned capacitation, acrosome, binding and binding receptors, species recognition, the zona pellucida, vital envelope, acrosomal reaction, polyspermy. We've already looked at those terms. So you need to they're just a brief sort of guide. There's some terms you really have to know what they're doing or you won't know what's going on. So we'll pick up now with the fast block to polyspermy, slow block, and a little bit of other stuff here in a bit. All right, this slide right here, this is showing electrically the first type of block to polyspermy. It's called fast block. I'll write it up here. Fast. Terrible. I need to get a tablet I can write on because writing with this mouse is terrible. Fast block. Terrible letters. All right, there you go. If you have a 3-year-old at home, they can outright me. Fast block to polyspermy. All it is, when that first cell contacts the egg cell membrane. Now, I don't know if you looked at it last semester. We'll certainly look at it this semester. But cells have an electrical potential or charge across the membrane. Typically, it's about negative 70 millivolts. That's what they're giving you here. We'll look at the mechanism when we start the nervous system, but basically there's an unequal distribution of ions in most cells. There tends to be an accumulation of negatively charged ions inside, more positive ions outside. That gives you this electrical difference. If you plug in a voltmeter, you can detect that electrical charge. That's in millivolts, so it's pretty obvious. Most cells are running negative 70 millivolts. But look what happens here. So this is recorded by sticking an electrode in the cell and one just outside the cell membrane. So the membrane would be between the two electrodes and recording that electrical potential. Right as that first sperm enters the egg, this point right here, look what happens to the membrane potential. It goes from negative 70 to just above 0. That is referred to as a depolarization. We'll see this in many excitable cells, nerve cells, muscle cells. It's pretty common. But this is a signal. This change in membrane potential electrically temporarily, think of it as like stuns the sperm. It makes them hesitate. So it happens, in very short period of time. And after fertilization is here, it depolarizes here. We're talking, less than a second. Boom, it's depolarized. That inhibits the sperm temporarily, and they call that fast block to polyspermy. It blocks any sperm from entering momentarily, and it happens really fast, hence the name fast block. Now, I will tell you, fast block to polyspermy, we used to think it occurred in all the eggs in animals. It doesn't. When they started looking at placental mammals, we don't have this. We don't have fast block. We just have slow block. But that is fast block to polyspermy. It is just a depolarization, a change in the electrical potential across the membrane that temporarily inhibits further sperm action. All right, what we're going to look at here, this is sort of the next step. After fast block to polyspermy, which just takes a second or a fraction of a second. It's very quick, but it's kind of temporary. This next thing, you'll notice it takes a whole lot longer, 30 seconds or so. This is, surprisingly enough, called slow block to polyspermy. While it's a little slower to initiate, it's more permanent. Now, what they're showing you here, again, this is the outside of the egg here. This is the egg cytoplasm in here. A couple structures that I haven't mentioned yet. Right here, you've got some little membrane-bound packets. They're vesicles. They're along the outer edge of the egg, which is the cortical region or the cortex. It's always the outer edge of something that's called the cortex. So these are called cortical granules or cortical vesicles. Little membrane-bound packets that contain a mixture of chemicals in them, some enzymes and some other stuff. And what happens after fast block, when this cell depolarizes, it's due to the movement of some ions moving out, positive ions, and potentially ions moving in. Calcium's a common one. It's positively charged, moves in. That acts as a signal, and it causes these cortical vesicles to move and migrate towards the egg's plasma membrane, and they will fuse with it and release their contents into this space out here. This kind of area shaded in green is where the contents of the vesicles will be released. Notice that is between the egg's plasma membrane in brown, down at the bottom here, and the vital envelope up here. Now also note, I'll put some little marks on it, there are some proteins that actually connect the vital envelope and the egg's plasma membrane. So they're physically attached together, almost like one. That's when you look at an unfertilized egg, you really can't resolve the vital envelope and the plasma membrane. They're pretty much fused together on a light microscope. You can't see much. But what happens when the contents of these vesicles are released, I'll use a little eraser here, it basically breaks down the protein connection holding the vital envelope to the plasma membrane. So they're no longer fused. In addition, the contents of these vesicles, we'll kind of use that as area shaded in green, they're concentrated, they're hypertonic. That is going to cause the osmotic influx of water. That's kind of what this line right here is trying to represent, the osmotic influx of water. That water moves into the space between the vital envelope and the egg's plasma membrane. And you'll notice, on this diagram, you'll see how they're trying to represent them moving further and further apart. There's a pretty big gap here between the vital envelope and the egg's plasma membrane. So they're pushing them away. Well, keep in mind, all the rest of those sperm, they're out here somewhere. Let me in. Let me in. When you push that vital envelope away, you're basically pushing the rest of the sperm with it. That helps a bit. And then finally, you've broken a connection between the egg's plasma membrane and the vital envelope. You osmotically kind of fill up that space with fluid, so it pushes them apart. And then chemically, the contents of those cortical vesicles, they tan or link the proteins together, and they convert the vital envelope into something referred to as the fertilization membrane or envelope. That has been chemically altered. So now all the enzymes in the acrosome, it doesn't matter if you dump them on that fertilization envelope. It's inert. It's not going to be affected by those enzymes. And also no more sperm can get in. That is slow block to polyspermy. It takes a little longer to occur, but it's more permanent. Once that fertilization envelope forms, there basically aren't going to be any more sperm getting in there. No more polyspermy. All right. And this is showing you the actual eggs we'll be looking at. So these things here, I'm trying to outline it a bit. That is a sea urchin egg. Actually, it's a zygote. This has been fertilized. So you can tell by looking at it. The egg has this homogeneously mixed yolk in it. You can't really see many structures in it easily. A little bit of maybe nucleus there. But you notice this clear halo on the outside. That is a fertilization envelope. As soon as one sperm, and keep in mind, all these little black dots you see out here, is about 3 of them right there. Those are more sperm. They're still trying to get in. But once that first sperm fertilizes the egg, it pushes out that vitelline envelope and it hardens it to become the fertilization membrane. No more sperm can get in. And you can see this when we're watching the sea urchin eggs. We put the sperm in, put the eggs in, you watch, and after a while you see this little membrane, just like a balloon, go boop around the cell, and that's it. That's the fertilization membrane. So I know these 3 things, there's one there, second one there, third one there, those are zygotes. Those are fertilized eggs. So I'll stop there with fertilization. That's enough time, and I'll post this up. And then we'll go on to the next stage of development, which is cleavage.